Okay, so let's get this up. Yeah, going to look at photosynthesis. We sort of looked at it in 4 sections. The reason that we look at it in 4 sections is because basically up until now you've sort of just always done the equation, which probably come up here. Yeah, I'll pop it up here. This is the equation you've probably always seen, and it's we've always really basically simplified it a lot. We've always sort of said, okay, well, photosynthesis is the process where light energy is used to convert carbon dioxide and water into oxygen and glucose. And that's sort of as far as we've gone. So we've always gone, okay, plants produce their own food, their own energy. They don't have to eat or consume anything. And it does this through the process of photosynthesis. And that's sort of where we've always sort of gone, okay, and we've sort of left it at that. And then obviously we've got the word equation and then the formula equation. And we've basically just stopped there, and the symbol equation is what I meant. And we just sort of summarize it as that. Basically what we do now at A level is we sort of break it down and we actually go, okay, well, we know it happens in the chloroplast. We always say, oh yeah, photosynthesis happens in the chloroplast. It's where carbon dioxide and water becomes oxygen and glucose. And this is caused by light energy. And we don't really go into any further depth. What we're going to look at now is the structure of the leaf, how the leaf works and lets the plant do photosynthesis, what all the different chemicals it makes are, and then actually look in depth at the stages and actually what chemically is happening, because it's actually a lot more complicated than this, and actually a lot more, what's the word I'm looking for? A lot more products are made. It's not actually just oxygen and glucose, a lot more different molecules are made. But it's a good place to start. This is a good place to start to sort of recap ourselves. Okay, yeah, this is what photosynthesis is. But actually, it is a lot more complicated. So it's something I'm going to take probably quite slowly. And we'll go into a bit more depth. And it's one of those ones that people sort of, first time they hear it, sort of feel a little bit overwhelmed by it and sort of panic a little bit and feel like, oh no, this is quite a lot of information. And it is, and it's one of those ones that actually, the more you go back and look at it, and there's lots of cycles in it. So this picture will be probably back to front when I show it you, but I always have one jotted out in front of me when I come to teach photosynthesis, but there's like a cycle that looks like this. You basically just have to learn that. And I know I'm saying basically just learn a cycle of all these chemicals, but it's very repetitive. And actually the best way I would say to learn the different parts of photosynthesis at A level is actually just to write it down again and again and again, and then come back to it at another point and write it down without any notes and see how much you can remember. Because we can draw it as these diagrams, as these cycles, and I'll show you some in a bit, they're actually quite good ways of testing ourselves on, can we actually remember this? Because we can look back at them at a later date. We can actually get them back out and go, oh, hang on. No, I've missed this bit off, I've missed that bit off. So yeah, people do worry themselves over it a lot, but there's no point getting worried about it. It is very simple, it's very methodical, it's very much this happens and this happens and this happens and this happens. But there's just a lot of stages and you just have to know it in quite a bit of detail. So yeah. And when we get to those parts that we need to know in detail, I'll show you a few different ways that you can draw it and write it, because different people prefer it in different ways. So yeah, photosynthesis. So it's actually a 2-step process. And the first step is the light dependent reaction. And the second part is the light independent reaction. And this all takes place in the chloroplasts. And like I said, the end products aren't just glucose and oxygen, but actually we've got a complex organic molecules being produced, such as carbohydrates, amino acids, lipids, and nucleic acids. So we'll go into that. But basically this image here is our summary of this complex two-step process. So we'll have a quick recap as to why on earth plants bother doing photosynthesis. It's an essential biological process. It does this because it produces those complex organic molecules needed for growth, other than just glucose and oxygen. So if we go back, it makes those carbohydrates, it makes those amino acids, it makes lipids and nucleic acids. It makes all these complex molecules that we need for growth, what the plants need for growth. And it is also essential because it produces energy for processes in the organism. So it is also that form of energy that the organism needs to grow. And also produces oxygen, which is then used for respiration. But photosynthesis isn't just advantageous for the plant itself, it's actually great for other organisms in the environment. So this is also quite important to remember. So if later on down the line that plant is eaten, those organic molecules that we've spoken about, like the carbohydrates, nucleic acids, lipids, they are used to provide energy to the organisms higher up the food chain. So basically any organism that comes along and eats that plant, it gains those organic molecules into its body as well. So photosynthesis helps in that way. Also, the oxygen that's produced is released into the atmosphere and is available for other organisms. So for example, we need oxygen that we breathe in to use for our process of respiration. So therefore, the plants producing oxygen is also beneficial to us and any other animals. So that's why, whilst plants don't do photosynthesis to really benefit all of us, there is a lot of benefit from it as well in that manner. So photosynthesis, we know it happens in the chloroplasts. We know chloroplasts are found in the leaves of plants. You will have done, we do it in year 7, we do it at GCSE, looked at the parts of the leaf. You have to know that's still at A level. What I will show you, I think there's like a couple of quiz questions that come up on here actually, which are quite good, is that actually not only do you need to be able to label the parts and say, oh yeah, this part does this part does that, so the palisade level does X, Y, and Z. You also need to be able to label them from actual images of a plant. So what I mean by that is microscope images of a leaf cell cross section. So there's a couple of these coming up on here that I'll show you then. They're quite good. So they do come up a lot on the exams, and you'll get a photo or a drawing, and then you have to label them that way. Drawings obviously a lot easier because that's what we always get in textbooks. Again, it's quite good to look at lots of different diagrams of leaves because they all draw them slightly differently, so then it familiarizes yourself with sort of different drawings of them. So for example, we've got this one here that looks like that, and if I just make this a bit smaller for a second, I've got another one here, I'm sure I have. Yeah, I've got another one here. Roughly exactly the same, but it does look a little bit different. So it is sort of good choice to not just use one textbook or just use one revision guide, because what can happen is you go to the exam and they might draw it slightly differently. I don't mean like structurally differently, but things might just be spaced out a bit differently. If you're really used to just seeing it one way, it can sort of throw you. And then also looking at those real life photos. So we'll show you some of those in a bit. So quickly go through the different layers of the plant, the leaf. Won't really here at this point be anything massively new to you, but we'll go through each of the layers from the top to the bottom. I say from top to bottom, it's not actually necessarily following that exactly. So first layer that we always refer to is this top layer, and it's not actually a cellular layer. This top layer is called the waxy cuticle. So this area is the waxy cuticle, and it is a non-cellular layer, and it's on the surface of the leaf. You can get asked questions about, oh, what is the function? What's the purpose of it? And it's basically to limit that water loss. So it's got The reason it's called a waxy layer is it's got this basically, it's a waxy chemical particles on there, and they prevent water loss. So it's to stop transpiration occurring. It's also really, really useful because it's basically waterproof. So what's really good is if you think about, I've got a plant here, actually, I'm going to use this. If water falls onto the top of the leaf, like on this plant, it's got this waxy epidermis on it, this waxy cuticle. Because it's waxy and waterproof, what should happen is water can run off it and it prevents damage to the leaves. So basically this plant's even better shaped because it's actually got these curved grooves. And so basically what would happen is the water will sit in it in a different way and it stops basically the water gathering. If too much water gathers on top of the leaf, what will happen is it can basically either end up breaking the leaf off, so the stems can snap because of that excess weight. What's also great is if the water runs off the leaf, it should get excess going to the soil near to where the roots are for it, so it can take an extra water that way. So yeah, so the waxy cuticle, the main purpose of it is to stop that water loss, so to reduce the chance of transpiration occurring, but it also has that benefit of allowing water to run off it, which prevents damage to the leaves, and also actually, because it runs off and should get straight to the ground, it also helps the water to come up by the roots as well. So that's top layer, waxy cuticle. Then we have the upper epidermis. So this is our first cellular layer of our leaf. So the primary functions of the upper epidermis are basically just to protect your leaf from injury. And again, they also reduce water loss. So they are epidermal cells across the top. They have a nucleus in them. They're basically your very basic plant cell structured cells. So when water is going to come and sit on that top of that leaf before it runs off, they're that extra layer that protects everything in this parenchyma tissue layer, which is your really, really important layer that's got all your things like chloroplasts and palisade cells. It protects them. So it's basically really good. And it's another protection layer. Again, it's reducing water loss to help your plant stay nice and healthy. So yeah, it provides protection for the chloroplasts in the palisade cells as well from bleaching in bright light. So basically, the idea is chloroplasts need light energy for photosynthesis to occur. But if the sun is too bright and the light is too bright, it can basically bleach them and damage them so they wouldn't be able to work properly. So this upper epidermal layer provides physical protection, basically from things landing on it, water sitting there, provides protection from water loss, and also an extra protection to the chloroplasts to stop them from bleaching in bright light. So whilst they want light, it doesn't want too much light. It's a bit like us really. We need sunlight to be able to get enough vitamin D into our bodies. We get a little bit from eating, but not really that much. And sunlight provides a lot of vitamin D for us. However, if we went out in the summer and we're out in the sun all day long, I would get really, really badly sunburned because the sun is too strong for my skin in the summer and I would get burnt. So it's that sort of balancing act of, yes, we need the sun for vitamin D, but actually not too much sun. So it's like that with the chloroplasts in essence. So yeah, so we've got waxy cuticle, upper epidermis, and then we have the palisade cells. So our palisade cells, you may find in some textbooks them referred to as palisade mesophyll. Either is fine. And these are part of the parenchyma tissue. So the parenchyma tissue is made up of the palisade cells and the spongy mesophyll, okay? And these make up the internal tissue of the leaf. So that parenchyma tissue is internal tissue of the leaf, made up of palisade cells and the spongy mesophyll. The palisade cells, they contain a large number of chloroplasts, and they are adapted for efficient photosynthesis. We're actually going to look at the structure of chloroplasts in a lot of detail in a moment. So it's why we don't really know much. We don't really say much more about palisade cells other than going, there's chloroplasts in them. a thing that is quite important to take into account is the positioning of them. And this also helps when you're coming to label parts of the leaf structure from images, is if you look, these palisade cells here, they're towards the top. So again, the upper epidermis is going to block them from too much sunlight to stop them from getting bleached, but they're near the top. They're not near the bottom because the sunlight, that would be way too much thickness for the sunlight to be able to get through to them. So it's this layer here. Next layer, we're going to just skip the spongy mesophyll for a moment, and we're going to just jump all the way down to the lower epidermis. So our lower epidermis, bottom of the leaf, kind of self-explanatory in the name of it. Some of the cells in the lower epidermis are modified from guard cells. So you can probably remember a huge important part of the leaf is the stomata. Stomata are stoma is the plural, stomata is the singular, I think, or it's the other way around. I can never remember. I know that's really, really bad. I'm really bad with my scientific plurals, which way around it is. It's like aquaria and aquarium. Aquarium is a singular and aquaria is if you have lots of them. And I can never remember this. I have to write down some of them. So I apologize now. But yeah, guard cells, plural, so stoma I'm going with is lots of them. And one is a stomata. So back to what I'm actually talking about. Sorry. So stomata are basically pores in the cell that let gases in and out. So it's for gas exchange. Again, water can be lost through them. So the guard cells open and close. They open and close to control the movement and water and of water and gases for photosynthesis. So basically, if it were open all the time, if these pores were present all the time, too much water would be lost from the plant and it would end up dying, especially in hot weather. So guard cells are able to open and close to let gases in and out of the leaf, and also controls the water levels as well. The lower epidermis also works in a very similar way to the upper epidermis, and it provides that layer of protection as well. It's also a protective layer with these stomata in them as well. And so yeah, they allow the gases in and out. And then finally, we have the spongy mesophyll layer. The spongy mesophyll forms part of the parenchyma tissue of the leaf. These cells contain chloroplasts as well, but they're in smaller abundance than in the palisade cells. If we think about what I was saying about the light needing to get through and the wavelength of the light getting through, the distance that the light would need to travel from the top of the leaf to here would be a lot further than just getting from the top of the leaf to here. So there's not going to be as many chloroplasts in this level in the spongy mesophyll, just because it's not really economic for the plant to bother making loads of them. To make lots of chloroplasts there will take lots of energy, takes lots of space, and isn't necessarily beneficial to the plant. So they're not going to bother making loads, but there are some, because sunlight is going to get down there. So it's one of those balance off, it's a trade-off. We will have some, but we're not going to have too much. So yeah, this spongy mesophyll forms part of the parenchyma tissue along with those palisade cells. They contain some chloroplasts, but not as many as in the palisade cells. And then they have air spaces between the cells, and they're really important. If you think back to gas exchanges in leaves, we talked about transpiration before. The gas needs to be able to move around these cells. So we've got lots of this air space in there, and that's that gas exchange. So that is our basic structure of a leaf, and we will look into it in a bit for, we'll look at some of the diagrams in a bit actually for how they look a bit different with a real image. What we're going to look at now in some detail is actually the structure of a chloroplast. And this is something you'll never have seen before. Normally chloroplast is just sort of drawn as this like circle, or a bit similar to mitochondria. Sometimes people draw them, and that sort of it. We go, oh yeah, they're green. We find them in the palisade layer and a few in the spongy mesophyll, and that's it. But actually, now we need to know them in quite a lot of detail. So this is a chloroplast. Chloroplasts are an organelle. So we find them in a plant cell. So they're not a cell, they're an organelle, they're part of the cell. And they contain an internal system of membranes and pigments. So they are quite complicated. So this is one that's sort of been cut open and we're looking inside them. You can see that sort of similarity with the mitochondria here, which does sort of make sense because it is going to produce ATP. So whilst it doesn't work in the same way, it does have some similar structures to it. But yeah, so this is our chloroplast. It is an organelle, it isn't a cell. So that's really important to remember. So let's look at the different parts. Okay, so we have got, and you can probably see them quite clearly as we've opened it up, these stacks of discs. They almost look a bit like your vertebrae in your spine, that sort of stacked up upon each other. Each individual disc, so on this image we've got one highlighted here, so one individual disc is called a thylakoid. And this is probably the main thing that I've always found when teaching, that people get confused, is between thylakoids and granum. Thylakoid is one individual disc. The granum is the whole stack. And I'll repeat that again when we get to the granum, because it is one of them things that people go, ah, which one's which? So yeah, so thylakoid is one individual disc. Here it's classed as a flattened sac. You can call it a disc, it doesn't really matter. So they are pigment containing flattened sacs, and they're approximately 5 micrometers in diameter. So basically these are what make the chloroplasts green. That first, so if we think back to the beginning of this, I've said that photosynthesis is a complex 2-part process. The first part of photosynthesis, the first stage of it, is called the light dependent reaction. So it's the part of reaction that needs light for it to occur. And that takes place in the thylakoid membrane. So inside the thylakoid, that is where this light dependent reaction is taking place. So if you look at this whole chloroplast as a whole, if you look at how many stacks of them there is and how many individual ones, it's happening a lot across the chloroplast all at one point. So yeah, so that's what a thylakoid is. It's a flattened sac, pigment containing sac. So that's what makes it green, so the green, where that light dependent reaction takes place. Then we have the granum. Now the granum is the whole stack. So one stack is a granum. This one is a granum. And then if we talked about these three, we would call them grana. So if we're saying, oh, there's multiple grana in a chloroplast. So yeah, so a granum. And a granum is a stack of thylakoids. So it's all the thylakoids in that stack. A single chloroplast has approximately around 50 grana. Obviously, some will have more, some will have less, but that's how many they have roughly. These grana stacks provide the chloroplast with an increased surface area for photosynthesis to occur in a limited space. So basically, if you think chloroplasts are really, really small, that thylakoid was only 50 micrometers in diameter. If we stack all these thylakoids on top of each other, we're increasing the surface area. It's that answer that comes up all the time in biology. Why is it like this? Why we stack them? Increase surface area. And it is that process again. We're increasing the surface area in something that's got a very limited small space. So we've got more photosynthesis can occur. When in doubt in biology, it's often to increase surface area. So yeah, we've got one disc is a thylakoid, which is our pigment containing flattened stack. sac even, a stack of thylakoids is our granum. And then we have a few other things. So let me get rid of that because that will confuse us. We can have an intergranal thylakoid, or sometimes, depending on what book you're using, they're referred to as stroma thylakoids. They're the same thing, but I just thought I'd tell you the name of both because, again, depending on what textbook, what revision guide you're using, they can give them different names. These are extensions of the membrane that form the thylakoid. So the thylakoid has a membrane around it, and these are extensions of that. And what these do is they connect one granum to another. These are the site of the light-dependent reaction again. So these are where the light-dependent reaction happens, and it connects them. So that's really important. It connects all those granums, and they are the site of the light-dependent reaction. So it's these little yellow tubes, basically, that are connecting them. And it's sort of in the name, intergranal thylakoid. So intergranal, in between the granas, thylakoid, because it is part of the thylakoid. It's the same membrane that the thylakoid's made of. So then we have two more parts of the chloroplast we have to know. We have the stroma. The stroma is very, very similar to cytoplasm in a cell, okay? So the stroma is an amorphous gel-like material which contains enzymes, okay? And this is where that second part of photosynthesis takes place, and this is our light-independent reaction. So this is a part of photosynthesis that doesn't need light. It occurs after the light-dependent reaction, and it occurs in this gel-like material that's full of enzymes. And these enzymes are what's needed for this light-independent reaction to take place. And the thylakoids are embedded throughout the stroma. So basically the stroma basically fills that whole, any of the gaps in the chloroplast where there isn't thylakoids. And then the last part of the chloroplast is the double membrane. So it is not a single membrane bound organelle. It is a double membrane bound organelle. So it's double membraned or envelope, as some people call it. And again, it controls what's coming in and out of the chloroplast, very similar to a double membrane around a cell. The inner membrane is extensively folded to form the thylakoids. So actually the thylakoids are coming from this double membrane. It's folded to make these thylakoids to increase the surface area. So let's have a look at some of the images. Leaves and chloroplasts. Here we go. So chloroplasts, as we know, contain thylakoids, and that's an individual. That's an individual sac containing pigment, and the membranes of which are the site of the light-dependent reaction, and the stroma is where the light-independent reaction occurs. So I'm just going to put this on for a minute. Here's an actual image of one. I'm going to make this bigger so we can see this. So this is a microscopic image of this. So if we have a look here, we can see this stack for the granum really, really nicely here, actually. So this granum stack is really nice and clear here. We can see it really nice with them all stacked upon each other. There's a few others as well. There's not just one. So we can actually see another granum here. You can see one there, and that's probably part of one there as well. So we've got, that's a nice big clear one there. So we've got a stack of thylakoids, which makes up the granum. So yeah, you could draw a line to any one of those individual ones there. Now these that we can see, I'm going to colour this one in red, that we can see joining them here and joining them there. They're those intergranal thylakoids. So they're the thylakoid membranes that are joining each of the grana together. I'm going to undo that so you can see that better again. We've got our double membrane here, which is labelled because we can see the two layers of the membrane. And then obviously the stroma, which is that amorphous jelly-like substance which contains enzymes. I think this should, I think it will zoom in a bit more. Okay, no, what it is doing is it is changing the light here to show the contrast a bit differently. So as we're changing this, we can see brightness. So we're taking away the colour, so then you can actually see it as it would be drawn as a diagram. So if you were going to draw this as a diagram in a textbook, it would look something like this. You add the colour back in and it looks like that basically. So that gives you actually a really good image of how it might look in a textbook. If it's just drawn, you've got your thylakoid. Okay, yeah, so that is leaf there. Looking at the chloroplasts as well. And then a leaf section. So yeah, here's another sort of, this is an electron microscope image of a leaf again. So will it let me, yeah, there we go. Let's make this one larger as well. So this is actually a cross section of the whole leaf. So here we can see we've got the waxy cuticle layer, and that is always that top layer. Our upper epidermis, and they are always, when we're looking at these images and we're trying to identify these cells, these are the very, very square. I know they're not perfectly square, but these are the more square cells, are a lot smaller. They're normally a double layer as well, as you can see here. And then we have the palisade cells, which you can see here. And these are very, very long, very elongated. So that's quite a obvious sign of them. This is a vascular bundle over here. We don't actually have to worry about them too much, but you get them all over the leaf, and they're to do with other parts of the leaf, to do with like the xylem and phloem, and they will appear as sort of circles, and they get really, really small and really tight, and then sort of work outwards that way. Spongy mesophyll layer, you can normally point that as out because it goes in the opposite direction to everything else. It's not really very uniform. It's sort of all over the place. And then obviously, yeah, we've got the upper epidermis here. So hopefully this should do the same with the image. So yeah, as we start to take away the colour, the drawing should start to appear. So then we can see what it would look like as a drawing. I'm just going to speed it up a bit. Actually, I'm going to go back to here so we can see that better. So yeah, so if we're looking at it here, we can see that waxy cuticle is just a straight line on a drawing. Upper epidermis, someone's drawn in the cells there. Palisade cells, like I was saying, they're long, they're elongated, they're thin. And then you've got your spongy mesophyll cells that are normally lying on the side. And actually, there's only a few spongy mesophyll cells, and then these are all our air gaps between it. Vascular bundle, and then your lower epidermis. Then if we took it all away, that's what it would look like as a drawing that you might expect to see in a textbook. Remembering chloroplasts are located mainly in the palisade cells, but we will get a few in our spongy mesophyll as well. So yeah, plant leaf anatomy. So this is your basic sort of textbook diagram, which you can get asked to label in an exam. So it would be 1, 2, 3, 4, and 5. When you watch it back later, you could pause this and have a go at labeling it yourself. It might be one that you want to pause and print off or something. I'm going to go through the answers now really quickly. So yeah, 1, as you can probably guess, is our waxy cuticle layer. It's that top layer. And again, you're looking at where the arrow is going to. It's going to the top. 2 is going to be our upper epidermis layer. It's this top layer here. Remembering that provides protection for the leaf. It prevents water loss. And it also provides protection to the chloroplasts to stop them from being bleached. So that's our upper epidermis. 3. Also, it sounds a really silly point to make out, but make sure you follow where the numbers are. Most people go, oh yeah, 1, 2, 3, 4, 5. And it's not, it's going round in clockwise position. And it's amazing how many times people lose marks on questions because they just don't look at where the numbers are properly. That's all. So yeah, so 3 is our lower epidermal level, lower epidermis. There it is. Again, provides protection, prevents water loss, and is also the site of the stomata. And the guard cells, so obviously these are guard cells which open and close, which allow the stomata to open and close. Layer 4 is our spongy mesophyll layer. We have a few chloroplasts in this level, but not as much as in the palisade level. Got lots of airspace to allow gases in and out, gases to be exchanged in relief. And obviously over here, we've got a vascular bundle as well, which will involve lots of the xylem and phloem. And then that leaves layer 5 up here, which will be our palisade cells. These are the cells that contain lots and lots of chloroplasts. So let's have a quick look. Yep, all correct. And here we go. Here we've got a coloured image of a microscopic image of it. So again, 1 waxy cuticle layer. Top 2, we can see that layer of cells across the top, which is our upper epidermis. 3 is going to be our lower epidermis. It's these lower cells here. Now, I did this earlier and I think it tries to convince me that I've got 3 and 4 wrong, and I haven't. 4 is looking at all these cells over here, which is our spongy mesophyll. A few chloroplasts, not many. Spongy mesophyll. And then 5 is those palisade cells across the top there. Now, are you going to try and tell me if I'm doing this wrong? Yeah, you are, and I haven't done this wrong. Yeah, it's trying to tell me I've got palisade cells and lower epidermis wrong, and I haven't. Just ignore that. There's no way they're the wrong way around. It's lying. Chloroplast. So you can get asked to label a chloroplast in an exam. So one is our double membrane. It controls what's coming in and out of the chloroplast, which is really, really important, as this is the site of photosynthesis. So we don't just want loads of different chemicals coming in now. 2 is pointing at that jelly-like liquid. That is our stroma. The stroma is that jelly-like liquid. That's the site of the light-independent reaction, second part of photosynthesis. It contains enzymes and basically is filling all of the space that the thylakoids don't. 3, we're looking at one of them long stacks. Those large stacks are granums. Well, that's a granum, lots of them, grana. And they're all those thylakoids stacked on top of each other. 4, we're looking at one individual thylakoid. And remembering thylakoids are those pigment-filled sacs. So they're going to be green. They're the site of that light-dependent reaction in photosynthesis. And then finally, 5 is that intergranal thylakoid. So these are those, basically the tubes that are joining granum to granum, and they're made up of the membrane from the thylakoids. Here we're showing it as only like 1 per granum stack. Actually, they'd be multiple ones. It knows they're right this time. And then we've got an actual electron image of this. So these are a little bit harder sometimes to look at. So if we look at one appears to be pointing at the very, very top outside bit of this chloroplast. So we'd assume that it's talking about the double membrane there. What I would always say with these questions is, if we're a bit unsure, is actually go through them all, and then actually if you change your mind later and go, actually no, actually I think this one, I think 5 is the double membrane, you can go back and change it. 2 is pointing here, and I'm going to just draw this on because actually this isn't really very clear at all. Never mind. So yeah, so 2 is pointing at one individual thylakoid up here. So 2 is a thylakoid. It's one of those individual pigment-coloured sacs, whereas 3 is pointing at the whole stack of thylakoids, which makes it a granum. Generally, again, when they're pointing at the granum on an image, they will put those brackets around it. So it's always a nice clue that it's talking about that the image, that arrow and that label refers to the granum rather than a thylakoid. They'll make it really, really clear. 4, right, so this is when I'm going to struggle because this image isn't very clear. I think 4 is pointing at that part, that tube that's joining the 2. And so obviously, as we can remember, the tube that joins two is the intergranal thylakoid. It's the thylakoid that's going to join each grana, which means 5 must be the stroma. Okay, there we go. So we have to remember what their functions are, other than the things I've just said. So stroma. The stroma is the site of the light-independent reaction. So the stroma is that amorphous jelly-like substance. It has enzymes in it, and it's the second part of photosynthesis takes place in it, and that's the light-independent reaction part, sometimes called the dark cycle because there's no light involved. The thylakoid is the site of the light-dependent reaction. So that's the part that needs the light. That's the first part of photosynthesis. Chloroplast is a green pigment that is, no, it's not. Chlorophyll is a green pigment used in photosynthesis, and a chloroplast is the organelle that's found in palisade cells of leaves. So that was our plant, parts of the plant overview of our introduction to photosynthesis. So that is not what I want open. Go away. So part 2, again, because it's more complicated at this point at A level, we're not going to go diving straight into the different stages of photosynthesis. We're going to actually look now at the molecules involved in photosynthesis, because again, like I said at the beginning, we've said for years, photosynthesis is that really simple process where we've got light. Ooh, what have I done? There we go. Oops. Erase all. I don't know what I just did there. So where we say we've got light, and we're going to make glucose and oxygen with CO2 and water. But there's actually a lot more molecules involved in photosynthesis. That's a really, really basic overview. And it sums it up nicely, but actually it doesn't go into enough depth. So we're going to look at photosynthetic pigments. And we're going to look at 2. The one that actually is involved in photosynthesis is chlorophyll, but we are actually going to look at some other pigments as well. I'll explain why in a minute. We're also going to look at how we can investigate them. So it's a bit of a sort of an overview of how we might do a practical element to look at photosynthetic pigments. Have another look at ATP. Now, I know we've looked at ATP before. We're going to look at it in a bit of detail again and look at some of the light-dependent reaction. So, photosynthetic pigments. The definition of a photosynthetic pigment is it's a coloured biological compound, and it's present in chloroplasts and photosynthetic bacteria. So obviously we say, oh yeah, all plants go through photosynthesis. We do, but actually some bacteria are able to photosynthesize as well. So they would also possess a photosynthetic pigment. A photosynthetic pigment is able to capture light energy for photosynthesis. So in plants, there are 2 types of pigments. So we have chlorophylls, which are those green ones that we refer to most of the time when talking about photosynthesis, but we can also get carotenoids as well. One sounds a bit like carrots. There's a reason it sounds like carrots, and they're found in carrots, and it's what gives carrots those orangey colours. So they are coloured because they absorb particular wavelengths of light and then they reflect others. So that's why they have that specific colour. So photosynthesis doesn't just absorb any light, it absorbs a specific wavelength of light and works off that wavelength of light.